1 in 2 people get diagnosed with cancer in their lifetime, and 1 in 6 people die from it every single year. Unfortunately, cancer is kind of like that uninvited guest that just refuses to leave. And so I thought that we should do something about it, which is why I'm here to talk to you about gene editing, aka giving cancer the metaphorical boot. A few days ago, I got caught in a rabbit hole of researching about gene editing and how it can be used to potentially cure diseases, when I found Casgevy and Lyfgenia, 2 genetic treatments approved by the FDA to cure sickle cell anemia. I mean, imagine that, curing a genetic disorder right at its source. But then I asked myself, what if we could apply the same principle to cancer? I mean, with gene editing, we have the power to tackle cancer in ways we've only dreamed about. And so before I get into my solution, it's important to understand how cells work. Our cells are like factories, and DNA is the instruction manual that they follow. But just like when I leave my phone with my little cousin for a little too long, when this code goes wrong, things can get messy, and sometimes this mess can create cancer. But our body has something called T cells. T cells are our immune system's special forces, and they work to find malignant cells like cancer and take them out. However, cancer is kind of sneaky. Cancer uses a disguise, a don't eat me signal that it sends to T cells. It prevents T cells from recognizing cancer as an actual threat, and it allows cancer to grow inside of the body without getting detected by T cells. And so my solution to the problem was to remove the CD47 gene completely, to remove its function so that T cells can actually recognize cancer. To do so, I had to use a tool that would allow me to make precise cuts in the DNA. And to do that, I used CRISPR. CRISPR is like genetic scissors and requires DNA, guide RNA, and a Cas9 protein to function. I'll get into the guide RNA and Cas9 protein a little later, but imagine editing a document. CRISPR does just that, but it helps you delete harmful parts of the DNA and without impacting the rest of the genome. And that's exactly what I had to do with CD47. But what I had to do was perform a gene knockout. This essentially means removing the function of the gene so that it will stop working. And to do that, I had to create a guide RNA. So a guide RNA is like a GPS, which I put into CRISPR. It tells CRISPR where exactly in the DNA I have to cut. And I had to put this guide RNA inside the Cas9 protein. And this Cas9 protein is kind of like a truck. It takes the guide RNA to the specific corresponding section of the DNA that I had to cut. And guide RNAs are essential because they make sure that your experiment is efficient and effective. I ran this experiment on benchling, and after the simulation was complete, I ended up with a more effective and efficient T cell that can actually recognize cancer and take them out. So why does this matter? Imagine a world where cancer can't hide anymore. Removing CD47 is a first step towards this reality. We can change the number from 1 in 6 people dying from cancer to almost 0 in 6 people being affected by it. We can treat cancer like a common cold with gene editing. We can change the lives of millions of people around the world, and we can revolutionize cancer treatment. Thank you.